Good evening, everyone. Very good evening. How are you all? I hope you all doing good. Um, so today we will do the leftover topic from yesterday, and then we'll continue with the facts analysis. That is your fluorescent cell sorted analysis. So let's start one by one with each topic. So students will join us later. So basically, last time we were discussing about southern blotting, where I have discussed with you how majorly this technique is dealt with, where we do the electrophorese of your DNA nucleic acid, and then we transfer them over the nitrocellulose membrane, and then after transferring of the nitrocellulose membrane, you can see them under the radio label nucleic acids, and you can see your respective bands for the same. So for the same, we watched this video, but this was not coming good. So maybe I can play this video again today. So you can have this. I think if I play here, they will work fine. So it was cutting last time. So this will not, I think today will be fine. So I will play now. This will not have any sound now because it has the subtitles will coming in the below. So your membrane is ready to expose, and then you can see the things how you would like to see in your autoradiogram. The same way, actually, your your northern blotting is done the same way. There's not so much change in that. It's just particularly the source here is your RNA, and we also add some ribonuclease into this. So RNA is extracted from the cell of interest, but precaution must be taken to avoid degradation of single-stranded RNA by ribonuclease, so which is found on the skin on glassware. So wear gloves, usually specially treated plastic and glassware, to avoid accidentally introducing ribonuclease to extract the preparation. Additionally, we also add this diethyl pyrocarbonate into that, DPC, called as, which inhibits your ribonuclease activity, and also baking at high temperature might destroy ribonuclease activity. So keep that in mind. So the same steps as we did in the southern blotting that we have saw. Initially, the buffer that we're using will be a little bit changed. The rest, all of the technique is same. Yeah. So let's see how this northern blotting is done and practically. So I will share these videos later. So because if I share it now, it will be make our streaming slow. So we do the formaldehyde agrosgel electrophoresis in this case. So first we have our RNA. Is formamide and formaldehyde and MOPS. These three reagents are added to that. So mostly all technique is same, all the steps are same, just the buffer are changed, a little bit of timings are changed here. Formaldehyde agarose gels have been used here and the buffer was the same. Northern transfer here is done instead of southern transfer, put over the nylon membrane, then hybridization is happening. So they are put for the incubation for 50 degrees Celsius for 30 minutes and we do this washing 3 times. Yeah. Be careful, put you can see this here, a chamber in front, because the samples might be radioactively labeled. So we should not come in front of them, and then we check the results by the exposing to the X-ray film. So that's it. So whenever you do, you have to chance to do your southern blotting and northern blotting. That will be way to do it. So I will share these videos later with you. Now we go to the next part, that is western blotting. So this is for the protein, to get to know what your protein and over the nitrocellulose membrane, and in this case we use primary antibody and secondary antibody basically. So basically we can see after running your protein sample over the gel and transferring them over the nitrocellulose membrane. So initially we will add primary antibody to a specific protein. Then over that primary antibody you will add secondary antibody, and then after adding the secondary antibody you can see the visible blue band indicates the location of the protein. So that's how you have to maintain this yeah, western blotting technique. So it has various uses in identifying of any like mad cow disease, bovine spongiform, encephalopathy, yeah, HIV test, to know about disease versus non-disease state, how things are. So it has millions of applications at the end. Yeah. So this will have video now. So it's basically that what we did yesterday, if you remember, we did this 2D gel electrophoresis with that professor. That was a very long process where we have isoelectric focusing and then we were running the gel over the SDS page. This is also the SDS page basically, but here we are running our protein with the primary and secondary antibody. That's the main difference. And most likely this is highly technique that we use in the lab and most likely to be used. So let's start.
In this video, you will learn how to perform a Western blot. A Western blot can be used to identify specific proteins in a sample and provide information about the protein size and relative abundance in the sample. First, fill a tray with blotting buffer. You will be using this buffer to equilibrate your gel prior to starting the Western blot. Next, remove the gel from the gel cassette using the opening key. Line up the arrows on the opening lever with the 4 arrows on the cassette to open the cassette. After trimming the top and bottom of the gel with a straight edge, equilibrate the gel in the tray with blotting buffer for 15 minutes on a rocking platform. Pre-soak fiber pads in blotting buffer so that they are thoroughly soaked. To make a blotting sandwich, obtain a container large enough to fit the gel holder and add enough blotting buffer until the container is filled approximately 1 centimeter deep. Place the gel holder cassette in the container with the black side down and immersed in the buffer, and the white side up and out of the buffer. Lay 1 fiber pad flat on the black plastic. Next, wet a piece of blotting paper and place it on top of the pad. Be careful to avoid any bubbles between the pad and the paper, and make certain the buffer covers the paper. Take the gel and carefully place it squarely onto the blotting paper. Again, being careful to avoid any bubbles between the gel and the blotting paper. Next, you will be applying a piece of nitrocellulose membrane. Remove the protective sheet from the membrane and wet the membrane with blotting buffer. Carefully place the membrane squarely on the gel. Avoid moving the membrane once placed on the gel, as proteins will begin to blot immediately. Using a roller, remove any air bubbles between the gel and the membrane. Place a second sheet of wet blotting paper on top of the nitrocellulose membrane. Place a second wet fiber pad on top of the blotting paper. Fold the clear plastic side of the gel holder over the sandwich and clamp it to the black plastic side by sliding over the white clip. This tight fit will squeeze the sandwich together. Insert the gel holder into the inner module. Make certain that the black side of the gel holder is next to the black side of the module. Place the inner module into the electrophoresis chamber. Add a frozen cooling unit and fill the chamber with blotting buffer to the level of the white clip on the gel holder. Place the lid on the electrophoresis chamber. Connect the electrical leads to the power supply, making sure the connections are correct, red to red and black to black. Turn on the power supply and run the blot at 20 volts. If a timer is available, set it for 2.5 hours. When the run is complete, turn off the power supply and disassemble the electrophoresis chamber and remove the inner module. Open the module and place it in a container filled with blotting buffer with the black side down. Starting with the first fiber pad, remove each layer until you reach the nitrocellulose membrane. As you remove the membrane, note that the proteins have been transferred from the gel to the membrane. Note that the kaleidoscope pre-stain standards have been transferred and can be seen on both sides of the membrane. You can also see that there are no longer any proteins on the gel. Immerse the membrane in 25 milliliters of blocking solution and incubate it for 15 minutes at room temperature on a rocking platform. Pour off the blocking solution. Add 10 milliliters of primary antibody. Incubate for 10 to 20 minutes on a rocking platform. The platform should be set at a faster setting to ensure constant coverage of the membrane. Pour off the primary antibody. Rinse the membrane quickly in 50 milliliters of wash buffer and then discard the wash buffer. Add another 50 milliliters of wash buffer to the membrane and let it wash for 3 minutes on the rocking platform at a medium speed setting. Discard the wash buffer. Add 10 milliliters of secondary antibody and incubate the membrane for 5 to 15 minutes on the rocking platform at a fast speed. Pour off the secondary antibody. Rinse the membrane quickly in 50 milliliters of wash buffer and discard the wash buffer. Add an additional 50 milliliters of wash buffer and wash the membrane for 3 minutes on the rocking platform on a medium speed setting. Discard the wash buffer. Add 10 milliliters of substrate. Incubate the membrane with the substrate for 10 to 30 minutes with either manual shaking or on a rocking platform. Watch for the color development. Once the colors have developed, rinse the membrane twice with distilled water and blot dry with a paper towel. Air dry for 3 to 60 minutes and then cover in plastic wrap for storage.
So that's how the final Western blotting technique is taken place. The same way now. So we have discussed now southern blotting, northern blotting, and western blotting. Now the same way you can do southern western, which will detect the relationship between your DNA binding proteins. So we can recognize the same method, same technique, and we can recognize with the southern western blotting to know what which DNA is binding to which protein. In the northern blotting, it will be instead of DNA, here will be the RNA interaction. So we will check northern in the protein RNA interaction. So which protein with which RNA they are being labeled. So the last, there is one dot blotting. It's actually modified version of a western blotting in which we analyze the protein of interest. And dot blot methodology differs from the traditional western blot technique by not separating protein samples but using electrophores. And last not the least, this is not being used in the day-to-day life actually, but this is good from your subjective perspective, from your college perspective. If the topic is coming in examination, that sometimes they say explain zoo blotting, yeah, so this would be an essential topic to cover then. So let's see. Within this, zoo blotting is basically based on the principle of southern blotting, in which genome of any organisms or two regions. There are two regions within this genome, that is coding and non-coding. And we get to know about the coding region that causes the interest or for most of the researchers. But the problem is that most of the DNA is non-coding actually. Around 90% is non-coding. Very few region which codes, actually 98%, only the 2% is coding. So we have to be very precisely when we are doing it. Extracted from a series of related animals such as human, monkey, mouse, hamster, cow, and samples in its DNA is called as zoo are cut with the like one restriction enzyme and then nylon membrane, and they probe with the specific human coding DNA. In all those things. So if DNA sample really does include the coding sequence, it will probably hybridize with the same fragment and with the closely related animals also. So we can recognize the coding region in another species also related to human. So for example, this is the known coding regions of itself in the human, and this is on the right hand side: rat, cow, dog, human. So we put our samples there, and then when you can see